Today the idea is to go over two of the more important cranial nerves, the optic nerve and the vestibulocochlear nerve. We'll concentrate on the vestibular portion of it for balance. We'll also talk briefly about audition, but first we're going to talk about the optic nerve. Did you all have basic, the eyeball and the semicircumference and A&P? Did you go over the special senses, what the eyeball is, what it looks like, etc.? Yes? You know where the eyeball is, what it does, right? Basically. Okay, so we'll go very, I don't care about all the physiology of this, but we're looking at the neuro aspects of the eyeball. So basically, light comes in the front through the pupil, right? We can regulate the size of the pupil, we've looked at that before. Hits the lens. Goes through the lens, hits the retina. Okay, simple as that. The key thing to remember about the lens, it's going to invert your image. Okay, so this is the eyeball and there's the lens. So let's pretend this is the images from the upper visual field. Okay, that is going to be refracted by the lens, it's going to go to the bottom of the retina. Okay, same thing, lower visual field information, hits the lens, goes to the top of the retina. Simple as that. Okay? So if I'm looking straight ahead at somebody, their feet are going to be in the top of my eyeball on the retina, their head is going to be on the bottom of my retina. Fair enough? Simple. But it also does it left and right as well. So now I could be looking at, let's pretend I'm looking at the top of the eyeball here. This could be the left-hand side, this could be the right-hand side. I'm looking down on top of the eyeball, right? So stuff coming in the right-hand side of my lens, the eyeball is going to be refracted by my lens and end up on my left-hand side of my retina. Okay, it crosses it, refracts it. Okay? So remember that. It's important that remember that the lens will invert the image left and right and up and down. Okay? So in other words, what you see in the world is on the opposite side of the retina because of the lens. Okay? We'll get back to why that's important in a minute. So again, light hits the lens, comes through the eyeball and ends up on this retina. The retina, of course, houses our receptors. Okay? As the optic nerve comes into the back of the retina, it's going to spread out and supply the retina with those neurons. But as it comes through the back of the eye, there's going to be a spot there where there are no receptors. There are no rods and cones. That's, of course, the blind spot. You've all heard that before, that terminology, right? Okay, let's look at the retina in a little bit more detail, very briefly. The top is where the light comes through. So we're looking at the bottom here of the retina. So the light's coming from the top of the screen, comes through these nerves, and basically hits the receptors here. Those are, of course, our rods and cones. I'm not going to go over the detailed physiology of how those work. I mean, you've had that before, I'm sure. But it hits the rods and cones, which then initiate that signal being sent to the brain. Okay, so the rods and cones are the things that pick up that light signal. And if you look at that, within the retina, there are there's a receptor and two neurons. There are three types of cells in series. Let me look at this here. Here we go. So in this case now, the light is coming from the bottom of the screen. Okay? The lens is at the top of the screen. The retina. So it hits the rods and cones. You can see the rods and cones have their own neuron linked to them. That neuron below those green lines, what kind of neuron is that functionally, do you think? Structurally, should I say? Bipolar. So these are bipolar neurons here. you can think of those as being the first order neurons, sort of. There's going to be 3 orders of neuron in this chain again to get to the cortex. 3 orders of neuron, but it goes different kind of pathways than your proprioception, whatever, obviously. You can see on the rods, though, many rods will synapse the same bipolar neuron, which then connects with these neurons out here. Okay? Let's look at that. So that corresponds to, there are bipolar neurons, which then synapse, take the signal to the ganglion neurons here. Those are also bipolar. You can see the cell body here. Okay, so we go receptors. Here, maybe another picture, same thing here. The grey neurons that are the receptors, the rods and cones. The red dots there are bipolar neurons. The G, the brown ones at the top are ganglionic neurons. Okay, the ganglion neurons make up what we think of usually as the optic nerve. Okay. So also, we often talk about the amacrine cells and horizontal cells within the retina. These kind of modulate what gets to the ganglion neurons. They kind of modulate, they mediate between the rods and cones and the ganglion neurons. The main thing is we've got our information now to our ganglion neurons. Receptors, rods and cones, bipolar neurons, out up to the ganglion neurons. The ganglion neurons then, those neural fibers, converge at the optic disc and leave the back of the retina. They're carrying our information about sight. And in that bundle here, this is optic nerves here, there's about 1 million neurons. There's about 1 million neurons coming out the back of each eye. Okay? They then they then leave the back of the eye and form the optic nerve. Okay? Right. Let's see what happens to that information. Okay, first of all we should probably go over this diagram before we follow it back. Okay. We can break our retina into 2 fields, left and right. Okay? The retina that is next to my nose, the medial side of my retina is known as a nasal retinal field. It sits next to my nose. So that corresponds to C and B on this diagram. Okay? These are retinal fields. So if I was to look at an eyeball, a retina from the top, let's say, so here's the retina, and here's the light. Okay? This is the bottom, looking down somebody's head. And here is their nose. Okay? We're looking that way. This is obviously this is the person's left retina. So we can split that retina into 2 retinal fields. The half that is nearest the nose is known as the nasal retinal field. The half that's nearest the temporal bone is going to be the temporal retinal field. Okay, so you've got a retinal, 2 retinal fields. One next to the nose, one next to the outside of the eye. Temporal and nasal. Fair enough? Those retinal fields see the opposite visual field. The visual field means what's out there in the environment. The visual field. Okay? So if we look at this green line here, this part of the retina here, the left eye's nasal retinal field sees this temporal visual field out here. That makes sense? The inside of my eye sees the outside of the world. The outside of my eye sees the opposite side of the world. Sees past my nose because of this refraction of the light of the lens. Does that make sense so far? Two retinal fields see two different visual fields. And always remember, when you refer to a visual deficit, you always refer to the visual field. You never refer to the retinal field. So when you see diagrams of visual cuts, visual deficits, those diagrams will show you the visual field that the person can't see. Never think of a retinal field damage, lesion. Always think of the visual field problem. Okay. This diagram also shows that when I'm looking straight ahead, okay, everything to the left of center is known as my left visual field in green. Everything to the right of center is my right visual field. Simple as that. Alright? Okay, that left visual field is seen by D and B on this diagram. Okay, those green lines, D and B. In other words, my left visual field is made up of my left eyes, or is seen by, let's say, seen by my left eye's nasal retinal field and my right eye's temporal retinal field. They look to the left. The right-hand side goes to the opposite. So my right-hand visual field is seen by the outside of my left retina, which is my left temporal retinal field, and the inside of my right eye. My right eye's nasal retinal field sees over there. Does that make sense so far? Just follow this diagram, it's pretty clear, hopefully. Does that make sense? Retinal fields see opposite visual fields. Okay? So, this is important to remember because when we get this stuff sent to our visual cortex on the back of the head, the occipital lobe, right? It's not the left eye that goes to my right hemisphere. It's not my right eye that goes to my left hemisphere. It's the left visual field goes to my back of my right hemisphere. It's the right visual field goes to my left hemisphere. Make sense so far? Okay, let's follow that optic nerve back then. So these red and green lines here, I made this diagram, hopefully it's helpful. That's my optic nerve. That corresponds to the optic nerve here. Okay? The optic nerve leaves the back of the eye. This is probably as good a diagram as any, this thing I made up. Okay. Then the optic nerve goes back and hits the optic chiasma. Chiasma cross. That's where they cross. Well, there's an X shape there, right next to the pituitary gland, remember. Now, as you can see here, my temporal retinal fields stay ipsilaterally. The nasal retinal fields cross. What I mean by that is, if you look at this red line here coming off this left eye's temporal retinal field, it comes down here, hits the optic chiasma and goes ipsilaterally. It stays on the left-hand side. This green line over here stays on the right-hand side. But your nasal retinal fields cross. Okay, so just remember this figure. Just remember, do that. That's all you need to remember. That is the optic chiasma. Draw an X and do these two things either side. that representation tells you all what happens in the optic chiasm. You must remember that figure, that pattern, that relationship. And that will explain what goes where.
How come it looks like they all cross in that picture? Am I looking at it?
You're looking at it wrong then. So this line here, this is part of my left optic nerve, right? Seeing coming from my left temporal nasal retinal field. It comes down here, it does not cross the other side, does it? It bends and turns like this, yes? So it's staying the same side, it's staying ipsilaterally. Make sense?
Isn't that the nasal retinal field?
Oh, have I labelled it wrong? A is left eye temporal retinal field. A, yes?
Up top on
Up top it says nasal.
That's visual field. Remember, the retinal field is the opposite visual field. Yes? You've got to get that straight. Retinal field is the opposite visual field because of the lens refracting the light. Okay? So, again, your nasal retinal fields cross. But what do those retinal fields see? They see a temporal visual field, don't they? The insides of my retinas see the outsides of the world because of the light crossing at the retina, at the lens. Okay? So, this is how we get the left side of the world to the right hemisphere. Okay, the left-hand side of the world in green here on the top is made up of my left eye temporal visual field and my right eye nasal visual field, right? Where does that information go? The left eye's temporal visual field comes down here, hits the inside of my eye here, the retinal nasal field. Where does that go? It crosses at the optic chiasma to get to the right side of the brain. This green line hits this temporal retinal field. Where does that go? It stays ipsilaterally and gets to the right hand side of the brain. So it's the optic chiasma where the optic nerve desiccates, crosses and splits. Some stays ipsilaterally, some crosses. If it's coming from the other side of the world, on the outside of the world, okay, it's going to cross. Your temporal visual fields cross. Or, similarly, your nasal retinal fields cross. The same thing. But always refer to the visual world we talked about deficits, and we'll get to that in a sec. Does that make sense so far? The optic chiasm, you've got to understand that. You've got to follow it through logically, see what goes where, and it all makes sense. Okay. So you've gone through this optic chiasm, now you've got everything from the left-hand world over in the right-hand side of the brain. Everything in the right-hand side of the world is now in the left-hand side of the brain. And you can see it there. Another great diagram. After it goes through the optic chiasma, we change the name to the optic tract. Whatever. Optic nerve, optic chiasma, optic tract. These are all these same ganglion neurons we looked at coming out the back of the retina. They then, where do all our senses have to go through? What's the big relay station? Thalamus. So they end at a specific nucleus, a part of the thalamus called the lateral geniculate body, or the lateral geniculate nucleus, either way. Have I labelled it on this one here? Yes, here. Lateral geniculate body or lateral geniculate nucleus, either way. That's where that, in this case, it's like a second order neuron. Remember, our bipolar neurons first, ganglion neurons second, third order neurons start at the thalamus, the lateral geniculate nucleus or lateral geniculate body. Where do they go? Where is the visual cortex?
At the
The back of your head, right? On your occipital pole, right at the tip of the back of the head. So those fibers now, those third order neurons leaving the lateral geniculate body, there's various terms for those. One is called optic radiations. What are they called there? Optic radiations. Or geniculocalcerine fibers. Geniculocalcerine fibers. Same thing, optic radiations. They spread out and they go along through the back of the brain and end on the occipital lobe, right around the calcarine fissure. The calcarine fissure is a line, a horizontal line. You can just see it here. There. A better diagram. There, that's kind of a sort of a cartoon version on the bottom there. The calcarine fissure is a horizontal line, okay? But down the center is going to be what? What's the big crack comes down the middle that separates the hemispheres? What's this crack here? What's the crack that separates left and right hemispheres? Longitudinal fissure, right? Big crack right down the middle. So in other words, you've got a left hemisphere and a right hemisphere back here. You've got a left part of the visual field and a left cortex, right cortex. So you've got to split down the middle and you've got to split across. It's like an X, basically, across. Okay? So everything on the left hemisphere comes from the right visual field. My left visual field goes to my right hemisphere. Okay? Now that horizontal fissure, the calcarine fissure, separates two gyri. There's a gyri above that fissure and a gyri below. Gyrus. And they're known as the cunate gyrus on top and the lingual gyrus below. Sometimes you just see cuneus rather than cunate gyrus, cuneus. Cuneus means cuneiform writing. Cuneiforms in your foot. Wedge. A wedge-shaped thingamajig. Cuneiform writing, they write little wedge-shaped stamps they made. Okay. So, got it so far. So, the optic radiations now go back to this calcarine area. But the upper and lower visual fields go kind of different ways. Remember, the bottom of my retina is seeing what part of the visual field? The top. So if you were to look at the retinal image, let's say, here's somebody's eyeball, here's the lens, and they're looking at somebody, whatever, let's just make it. They're looking at somebody like that, okay? That image is going to be like that on the back of the person's retina, upside down. So these fibers now come backwards in the optic nerve like that, relaying that information. Okay? They keep in that same orientation. They go back through the optic nerve, through the optic chiasma, through the optic tract to hit the lateral geniculate body. Then they carry on in that orientation. The fibers from the lower visual field are going to be on top. So, in other words, if we look at the primary visual cortex, the back there, you're going to basically see, let's carry on back here. Here is now my visual cortex, let's pretend, that's the back of my head. Okay? Here is the calcarine fissure. You will see the same thing there. In other words, my upper visual field goes to the bottom. And my lower visual field, the person's feet, goes to the top. And this is the cunate gyrus, and this is the lingual gyrus. So in other words, the bloke's head, or my upper visual field, ends on a lingual gyrus, beneath the calcarine fissure. The bloke's feet, the lower visual field, ends up on the cunate gyrus, above the calcarine fissure. Make sense? And let's pretend I was staring straight ahead and there was a bloke over here and two people, one over the left, one on the right. The guy on my left-hand side is going to end up where? Right cortex. The guy on the right-hand side ends on my left cortex. And they will both be upside down, of course. Okay? So look at this diagram. This is important in that this is a lateral body, nucleus. These are the fibers carrying the lower visual field. They kind of go straight back. But these fibers that go to the, carrying the lower visual field, sorry, the upper visual field, the bloke's head, okay, they're going to make this sweep through the temporal lobe and then go back. And this big sweep here around the lateral ventricle, which is that grey area in the lateral ventricle there, is known as Meyer's loop. We should get a picture of that somewhere. So there, it's circled there, Meyer's loop. Or let's pull it out there, Meyer's loop, whatever. Not a very good diagram there, but
So that's information from the top visual field?
yes, it's going to go to the lingual gyrus. Upper visual field information goes through Meyer's loop. Slightly different anatomical pathway than the lower visual field. Okay. Got it so far? Image inverted left and right. Image upside down. Left visual field, right hemisphere. Right visual field, left hemisphere. Upside down the back there. Okay? Now let's look at some lesions to this optic system, this optic nerve, etc., etc. Okay, remember, if all visual deficits will be described in terms of visual field loss, not retinal field. Big mistake, people in exams will give me a retinal field loss. Do not do that. So typically what you'll see in a diagram is two circles, okay? These do not represent the eyes. These do not represent the retina. They represent the visual fields that those eyes can see. Okay. So if I was to do this, this is make up something. How would you describe that loss? Just in terms of what does that patient cannot see? It is the visual field of the right eye. It's the temporal visual field, isn't it? That's not the retina. Don't think of it as retina. Think of this as what can't this eye see out in the real world. So in other words, this diagram is slightly confusing here in that think of these two circles here, right, as being here. You could draw two circles on your diagram, right there and there. Not out here, here. So your circle there comprising this green and red line and green and red line there, that's what these two things on the board represent. So in other words, if I draw a circle here like that, you'd see this. Now what, if I blacked out this side of the visual field, so in other words, I blacked out this red line here, right? Makes sense? If I've got a circle here, it's the right eye's visual field gone. Don't think of those as a retina. Think of those as what can the eye see. Okay? Keep that in mind as we go through these visual deficits.
Would you say the right visual field of the right eye?
No, you would say the temporal visual field of the right eye. Temporal visual field. This will make more sense here. Well, or confuse you even more. Okay, if I cut your right optic nerve, what would you not be able to see? Two circles. What could you not see if I cut your right optic nerve? Would I lose my complete left visual field? Would I lose my left visual field? Yes or no? No. I have got to cut your right optic nerve. So I'm going to cut, I could poke, in other words, I'd poke your right eye out. Or you put a patch over your right eye. Same thing. I've completely destroyed the right eye. Okay? So in other words, I've cut here. Or if I take your right eyeball out of the picture there, could I see the left visual field? Yes, because my left eye's nasal retinal field looks over there. So if I close my right eye, I can see over there. It's this part of my eye seeing over here. Okay? Can I see over here? Can I see right eye, right visual field? Yes. But I'm not going to see over here because my nose gets in my way. Okay? So you've still got bits of those visual fields, but so how would you describe it? This is what you draw, wouldn't it? It's basically a nopsia of the right eye. You just do this. Right eye nopsia, simple as that. You lose a retina, you lose the eyeball, you use your right optic nerve, you've got right eye anopsia. Simple as that. And that's represented in the very top diagram here. This one up here. Okay? Okay. Then, once upon a time, you have let's go back to this diagram first. Now, look at the relationships of the pituitary gland and your internal carotids to the optic chiasma. When you do dissection in your 6th year or whenever you do it with Chris, you can see the optic chiasma right there, and you have your two internal carotids coming up here, right next to the optic chiasma. Behind the optic chiasma, when we looked at this earlier, is my pituitary gland. Problems in those thingamajigs can affect the optic chiasma. Okay, if we get an aneurysm in my right internal carotid, it swells up, we get hardening here sometimes, you will damage these fibers here. Only. You will damage the part of the optic chiasma that comes from the right eye's temporal retinal field. But remember what that sees. Remember what D is looking at. So you have, remember your two circles, draw your circles here, here and here. And you would get, obviously, if you think about it, yes? That's what you would see. Okay? Look where it's going. This green portion here, these green fibres here would not be able to see anything. There's your circle. The green is gone. Okay? And that is known as right nasal hemianopsia. Heminopsia, half blindness. Right eye. Why nasal? You're losing your nasal visual field, not your nasal retinal field. This is my nasal visual field. Okay? Would you notice that? Would a patient tell you, I've got right nasal heminopsia? No. But would they notice that perhaps? Maybe not. How would they notice it? How would you show it's gone? You close your left eye. Because, remember, look where it's seeing. Your left eye can see most of that stuff over there. A bit of it, anyway. So you might not notice that. So how would you notice this? You'd basically have them close their left eye and put a pen out here, pencil the finger and say, tell me when you can see my finger. And you go, yes, that's fine. Okay. Tell me when you can see my finger over here. Say, normally, with both eyes open, I can see this pen here, easily. Close my left eye and you say, when can I see that pen? I'll go, I'll do it realistically. There, I can see it now. Okay? If I have this problem, retinal visual field deficit, I'll go, tell me when you can see this pen. Now. I normally can see it, just as it clears my nose, there. As it comes past my nose, I can see it. But I wouldn't if I had this issue. It would have to be way over here before I can see it. Past midline of the eye, perhaps. Does that make sense? Because of your right internal carotid aneurysm. Okay, then one day you decide to have a pituitary tumour. Okay? You decide to, you want to try one. Okay? And you can see that blue thing that represents my pituitary gland, that swells up and pushes forward. And what this does is impedes, causes a lesion in the fibres that cross. So it cuts off the crossing fibres here that are coming from the nasal retinal fields. The nasal retinal fields see what? The outside of the world. So, what would be cut off in these two eyes then? What would I lose out of these eyes? Both temporal visual fields. Like that. That's gone. Okay? And that's the third diagram down there. Bitemporal, both temporal visual fields, bitemporal hemianopsia, half blindness. Like they've got blinkers or blinders on. Like a horse, wears the kind of basic, another term would be tunnel vision sometimes. Tunnel vision. They can see in the middle, they can't see, no peripheral vision, it's gone. So you give a test to that patient, you say, when can you see my fingers? And you do this, and they're about here. I can't see anything out here, so it's gone. Okay? And that's the third little set of diagram there. Now let's say we take out all the fibres in the optic radiations of the right-hand side of the brain here. Okay? So we've got a lesion here. You completely lose, it could be a, I don't know, it was the calcarine artery comes off the posterior cerebral artery. We'll get to that when we look at the cerebral blood flow, but the calcarine artery supplies this visual cortex. Okay? So you should probably remember that for now. So the calcarine artery goes to the visual cortices. With the right and the left one, of course. The right one goes to the right cortex, the left one goes to the left cortex. Calcarine artery. So if I had a right calcarine artery stroke that completely wiped out my right visual cortex, what couldn't you see? I've taken out your right visual cortex completely. What couldn't you see?
Your left visual field.
Everything at the left-hand side of the world. The left visual field is gone completely. And that is shown on the bottom there, bottom of those diagrams. Left, homonymous hemianopsia. Left, both sides, homonymous hemianopsia, half blindness. Can you read that there? I've got another diagram somewhere of that. Where it's labelled down there, right hemianopsia, left hemianopsia, whatever, whichever side it is. You're talking about the right visual field is gone. Okay, got that so far? Okay, next step. Remember this diagram. The visual field, the upper and lower visual fields are represented on different parts of the visual cortex. In other words, the lower visual field goes to the cunate gyrus. The upper visual field goes to the lingual gyrus. And the pathways go differently. This is the key to remembering this. Your upper visual field, remember, goes through Meyer's loop. Goes through the temporal lobe. The lower visual field does not. Okay? So, if I get a temporal lobe lesion, and there's many reasons why you might get that, and we'll talk about some of those later. If I get a, what did I say, left or right? Did I say? Okay, let's say left. I have a left temporal lobe lesion that wipes out Meyer's loop. What would you lose? Exactly. You'd lose which side of the world for a start? Left or right? Left side of the brain, which visual field, left or right, would I lose? Right. And because if I only taken out Meyer's loop, which part of the visual field will I lose? Upper or lower? Upper. So you'd get what's known as a quadrant anopsia. A quadrant, a quarter. And that would be the opposite of this. So this is on the right-hand side. But I said the left hemisphere, whatever. Okay? And here's probably a better diagram. Okay? So you're going to have, which one should we pick up? So you can have superior and inferior quadrantanopsias, coupled with left and right. Okay? If it's the right visual field, it's a right something quadrantanopsia. If you've lost the lower visual field, okay, it can be a right lower quadrantanopsia, homonymous quadrantanopsia. Okay? So let's look at what is that number there? Number 6. Number 6. This fella here. You can see that lesion is over here in the left-hand hemispheres, Meyer's loop. Okay? So it's seeing, remember, the upper visual field. So this is what the patient would report. You have to do this with a visual cut. You move your fingers up and down, one eye at a time, and you can see what they're missing. And they'll be missing this. Number 6. And that would be right, visual field, right, so it's right, homonymous, superior quadrantanopsia. Does that make sense? And you won't get it right now, just look at it. But if you look at these diagrams, it's pretty clear what you're going to see. Okay, I'm going to lose your right you're going to stroke something, and you're going to lose your right lingual gyrus only. The right lingual gyrus. So think about what I would lose. My right lingual gyrus. So, what does the right hemisphere see? Left visual world. What does my lingual gyrus see? Upper visual field. Let's lose the cunate gyrus on the right hand side. What would you see then? You'd use the left lower visual field. And I think that's... Is that on there? No, it's not. This one here is sort of showing 7. Where's that lesion on there? Okay, yeah. You can see number 7 here is losing the fibers in the lower visual field going here. That's the cunate gyrus. So you would have an inferior, homonymous inferior quadrantenopsia, and this would be the right side of the, so it would be right inferior quadrantenopsia. Make sense? This diagram probably, that diagram shows you everything. In terms of pathways. It's all there. You can see how the left and right fields go to the cunate gyrus, the lingual gyrus, but all these diagrams basically show the same thing. Good diagrams in a book and in your anatomy book. Draw that, learn to draw that, and you'll know everything about it. Cut anything on there, figure out what's missing. Remember, these circles represent the visual fields, not retinal fields. Don't think of these as the eyeballs. Think of them as what the eyeballs can see. Okay? So in a question in the exam, I've seen this in the board exams, people have just, the exam just shows you 2 circles with a black side here and there. And they'll say, what is this? What is the lesion? Where would be a likely lesion for this? What do you call this? Well, I might in the exam say, I'm going to give you a pituitary gland tumour. What is the likely visual deficit? And you would say bitemporal hemianopsia, and you could draw the tunnel vision thing. Make sense? But these are very diagnostic in terms of figuring out where the damage is on this optic nerve, optic tract, etc., etc. Okay, let's get to this diagram. So far, all I've said is that all our nerves in the optic nerve go to the optic cortex. That's not correct. Okay? About 1%, let's look at this stuff, shown there, 1% of those neurons coming along the optic tract do not go to the lateral nuclear body. They peel off and go to the brain stem. Shown there. And I added this diagram this morning. I've updated your slides to show this diagram. Let's go back to this one perhaps. Okay. So the optic tract comes out of the optic chiasma. 1% of those neurons peel off and go to the pretectal nucleus. The pretectal nucleus, which is, as you can see here, this is represented. Here's the pretectal nucleus. And what's SC stand for? There. What's SC stand for? The tectum? Visual reflexes? Superior colliculi. Superior colliculi. The pretectal nuclei within the superior colliculi. This is the area responsible for visual reflexes. Okay. So So what happens here, this is the area responsible for rapid reactions to visual, for light, for example. Okay, you can see here there's a light being shone into this person's right eye only. So you get a little pinpoint flashlight shine right in the person's right eye. Those fibers come to the pretectal nucleus. There is then an interneuron to the Edinger-Westphal nucleus. Okay? And you can see there's a link between both pretectal nuclei through the posterior commissure. In other words, the right pretectal nuclei tells the left pretectal nuclei what's going on. So there's a bilateral link. Left is linked to right linked to left, whatever. So you get this light information. Somebody shone a light in my right eye. It goes to the pretectal nucleus. There's a little interneuron that goes directly to the Edinger-Westphal nucleus. The Edinger-Westphal nucleus is the start of what? The parasympathetic fibers within the oculomotor nerve, cranial nerve 3. What that does, of course, as we've seen in the oculomotor nerve before, goes out, the preganglion neurons go out to the ciliary ganglion, the postganglion neurons go out to what muscle? The sphincter muscle of the pupil, shown on the left-hand diagram. So you shine a light to somebody's right eye, the right eye closes. Simple as that. But because of this posterior commissural connection, the left eye also closes, but not as much, typically. That's known as the consensual light reflex. So in other words, the direct reflex, shine light in my right eye, left eye constricts. Concentric reflexes, shine light in my right eye, my left eye shuts down a bit. Make sense? Direct reflex and concentric reflex. The left eye consents to shut down a bit when the right eye sees some light. Okay? If I wiped out your visual cortex, you have a massive stroke on the visual cortex and you are blind. Okay? So your two diagrams obviously would be the left eye can see squat, the right eye can see nothing. I shine a light in a person's eye, would the pupil constrict? Yes. The pupil will constrict because these fibers peeled off the optic tract before anywhere near the optic cortex, the visual cortex. This is a brain stem reflex. Okay? Very important. Why? You can tell if somebody's about to keel over or not. If this is not working, hopefully you've got an oculomotor nerve lesion. Hopefully you've got an optic tract lesion. But more often than not, what will you have? Brainstem pressure, cerebral herniation, bleeding in the brain, pressing on my brainstem, shutting those vegetative functions down. This is one of them. If your pupil does not react to light, you are typically in a mess. And that is a medical emergency. Those people who cannot constrict their eyes with light flashing in it, it's pretty serious. It can be.
And that can be because of pressure.
You hope it's not. Yes, but that's the most serious thing can have. You have a head injury patient, car accident, whatever, and you shine a light in the kid's pupil and it's not shutting down, it could be cerebral herniation. It might be just a temporal shock system. Hopefully it's that. But if it persists, it could be serious consequences because you don't want your brainstem to shut down. Your brainstem shuts down, you're dead. You can lose your visual cortex. You can be blind. You can be blind. You can lose all the cognitive functions. You can live. If you shut your brainstem down, you're dead. That's what keeps you ticking over. So you need to check that reflex in an unconscious person. If the person's unconscious, this reflex will still work. No problem. If the brain stem's intact and these pathways are intact. But if you're pressing on the midbrain, you're in a mess. This light reflex, of course, this same rapid reflex here, what pathways start at superior colliculi?
Tectospinal.
The tectospinal tract, right? The tectospinal tract. So if you get a big flash of light, well, the pupil might constrict, but what else is going to happen? The person's going to blink like crazy and close their eyes down, blink. A visual doesn't have to be light. If I come to somebody's face and go like that, they will blink. The eyes are shut down. And what muscle shuts those eyes down? Orbicularis oculi. What nerve is that? Facial. There's a link here between this pretectal area and the visual, and it's in the brainstem, and the facial nerve. The facial nerve shuts the eyes down quickly. Okay, now you get even a bigger. Somebody jumps out behind a door and goes boo or whatever at you. What does your head do? And sometimes they'll go like this, right? You flinch. Your arms come to protect yourself, something like that, or you whack the person, whatever. You know, you go like that. Your head jerks back. Your hands come up from this bright visual stimulus jumping out at you. That's your tectospinal tract doing that for you. Starts in the tectum, superior colliculi, gets a hand in front of the face quick and get away from that thing which gets, getting in my face. You don't have conscious perception of that. That's not something you go, oh, somebody's jumped out at me, in front of me, I'm going to think about that. Oh, yeah, I better get my hands up, I better move my head back. No, it's a very, very rapid brainstem mediated reaction. Okay? This is sometimes thought to be the mechanism of blind sight. Have you heard of blind sight? This is where somebody's blind, okay, but they can still see, as it were. So, for example, if I've lost my, it's like you say, okay, let me give an example. There's something on a table out here, and they've lost sight, okay? But you say, reach out and grab it, and they go, what are you talking about? I can't see anything. And they reach out, and they'll often be a good attempt at grabbing it. Blind sight is when there's rapid reactions. You can see things, as it were, but not consciously. Okay? Because you do not get any conscious perception. It's not going to the visual cortex, but the brainstem can see, as it were, somewhat. It's a very weird phenomenon with blind sight, where somebody who's blind can actually see things reflexively, sort of thing. Another way of testing the ocular motor nerve, of course, we mentioned before, ciliospinal reflex. Remember the ciliospinal? What does that do? When you pinch somebody's face or neck hard, the pupil will what? Dilate. You're testing the ocular motor nerve there. When lots of things will affect your pupil. You take an exam. You can measure the amount of cognition, how much mental effort somebody's exuding using the pupil. The pupil will dilate as you get stressed out. As you think harder, the pupil will dilate. What drug dilates the pupil? Atropine, belladonna, etc., etc. You stick it in the eyeball, dilates the pupil. Why would a doctor want to look into the back of your eye? Check for herniation. You'll use an ophthalmoscope, right? An ophthalmoscope will look into the back of the eye and look at the retina. If you have cerebral herniation, swelling in the brain, you'll get what's known as a papilledema. A papilledema is also known as a choked disc. A choked disc. Choked disc. This is when the optic disc, the back of the lens, back of the retina, shall I say, this optic disc here, because the optic nerve has extensions of the subarachnoid space. The meninges come around the optic nerve. And this will push the optic disc into the retina at the back. You have a swelling. Edema, of course, means what? Swelling, right? I mean, you spell it differently, don't you? You don't put the O in it, is that right? Americans don't put the O in edema, is that right or not? You don't. You remember edema, Oedipus, all that bloke, remember? Oedipus King, you know, the bloke who was killed by his dad and he married his mum and his dad. Remember that bloke?
Yeah.
Had his ankles tied, Oedipus, and he was left on the mountain and a shepherd found him. Oedipus means swollen foot. Oedipus, foot, Ed is swollen.
We don't put the O in.
You don't put the O in it?
No.
Well, okay, whatever. Papilledema is a choked, swollen disc. In other words, the optic disc pushes into the back of the retina. And you look at somebody's eye using an ophthalmoscope and you can see it. You can see that increased pressure bulging the disc into the back of the eyeball. Okay? Many drugs, narcotics, will cause pinpoint pupils, unreactive to light. That's why a police officer who suspects somebody's on drugs, heroin, morphine, narcotics, will shine a light and look at the patient's eyeball. If it's not reacting to light and it's fixed pupil, called what? What's that called? A fixed pinpoint pupil. What's a fixed dilated pupil called? Mydriasis, remember that one? Mydriasis. And a fixed pinpoint pupil will be meiosis. Remember that one? I've mentioned those things? Have I or not? Yeah? Meiosis, pinpoint pupil. A person who's on drugs, stoned out their mind on heroin, morphine, that pupil will be fixed and unreactive to light. That's a test that police officers will often use. That's one reason, for example, if you have suspected head injury, they should not have morphine. Two reasons, of course. Morphine causes more bleeding. If the brain is bleeding, you want to increase it. So morphine actually causes dilation of blood vessels and will cause more bleeding. If a patient's got head injury, that's the last thing you want to give a patient for pain is morphine, because you're going to make the cerebral herniation worse. And of course, if you give them morphine, it pinpoints their pupils, and you cannot tell the state of the brainstem if the pupils are just fixed. You can tell the state of consciousness. I can't, which is skill, but you know, an anesthesiologist will look at the pupils and keep testing them, and they can test how deep under the person is as they're going under for surgery. So the pupils are a nice window into the function of the nervous system, as it were. A couple more things. Not enough time to talk about the vestibular system either. Okay, try this. With a partner, get your finger and move your finger to your partner's nose like that. What happens to the eyes? Try that. Look at their pupils! Look at the pupils and look at the eyeball. Okay. 3 things should be happening. 2 you can see. Okay? First thing that's going to happen is this is what's known as the near point reaction reflex, or near point reaction reflex, it's called. So anything comes close to you, near point reaction reflex. First, which you cannot see, of course, is the lens is going to get thicker, isn't it? The lens is going to get thicker because you want to focus on the finger. You can't see that happen. Okay? That's called accommodation. You're accommodating to that approaching stimulus. Okay? You can't see that. Which muscle's doing that for you? Ciliary muscle is the thing that's controlling the shape of the lens, right? So your palatina fibers are going to allow it to sphincter muscle, and you're going to focus near on. Long sight, long focus is sympathetic. Okay? That's, you want to see a lot. If your sympathetic system is dominant, you're going to see long distances. If the parasympathetic system is dominant, that's used for looking at things close up. But you can't see that. Your eyeballs, hopefully, went inwards, didn't they? They converged. You went cross-eyed. What muscles are doing that for you? Medial rectus. Medial rectus, what nerve? What nerve goes to medial rectus? Oculomotor. So your oculomotor nerve is working, hopefully, if both eyes came inwards. And what happened to your pupil? Did anybody see any change in the pupil? It constricted. If you look carefully at the pupil will constrict. Why on earth do you do that? There's no more light, is there? It's not constricting due to light, it's constricting due to your finger getting close to the person's nose. Why is that? It increases the field depth of focus, isn't it? It increases the depth of focus. Like a camera, right? You know, the old SLR, when you can manually adjust the aperture, you want to have somebody here in focus and the background in focus, you'll shut the f-stop down, won't you? You make it smaller hole. A smaller hole increases the depth of focus. So the pupil is acting like a camera. It's shutting the shutter down, or the aperture, should I say, to make a pinpoint pupil. That will allow you to focus better. The eye's doing that for you. That's called the near point reaction reflex. There are certain diseases where a pupil does not react to light, but will react to this near point reaction effort thingamajig. So an object coming close to you, the pupil will shut down, but with accommodation, but it does not react if you shine a light in somebody's eye. That's known as the Argyll-Robertson pupil. The Argyll-Robertson pupil is when the pupil does not react to light, but it does react to accommodation, to a near point stimulus. You'll see that in certain diabetes sometimes, and our old favourite, neurosyphilis. You'll see Argyll-Robertson pupil, I think it was 1908. This bloke discovered this, Argyll-Robertson. 1908, honestly, I can't remember exactly the date, but that's on certain diseases. You'll see that. If you see Argyll-Robertson pupil in a note, whatever, it means something's messed with the nervous system. It will not react to light, but it will react to near reaction coming towards you. Questions? Right, now vestibular system, right? In 5 minutes.
Good.
Okay, we'll call it quits. Draw those diagrams. Draw the optic tract, optic chiasma.